am Dr. A.P. Patil. Dear students of B.Sc. First Botany, Semester 1st, Paper 2nd is Biodiversity of Archegoniate. In this unit number 2 is Pteridophyte. Today we will see about the Pteris. Classification of Pteris. It is according to G.M. Smith. Kingdom Plantae, Subkingdom Cryptogams, Division Pteridophyta, Subdivision Pteropsida, Class Felicini, Order Felicales, Family Polypodiaceae, and Genus Pteris, Species Vittata. Now we will study about the morphology of Sporophyte of Pteris. It is terrestrial genus growing in moist shady forest or hillsides. It is with root, stem and leaves. Roots are slender, black and wiry, arise from ventral side of rhizome. Stem is rhizomatous, underground, branched, perennial and covered with brown scales. Leaf bases are present on the rhizome. A leaf is macrophylous, unipinnate and imparipinnate and arise from ground rhizomatous surface. The rhizome is underground structure. Bipinnate leaves are seen in Pteris biurita. Decompound and digitate leaves are also present in the genus Pteris. This figure represents external morphology of sporophytic plant body, which shows leaflets, rachis, petiole, fertile leaflets, then margin of the leaf is reflexed, midrib is present in the leaves, forked veins are present in the leaves and sori are present near the margin. Let us start to study the different parts present in the plant. Petiole base is covered with brown scales and sometimes with ramenta. Ramenta are the hair-like structures. Rachis has several sessile lanceolate, coriaceous leaflets arranged in pairs. Middle leaflet are larger and the other leaflets show gradual decrease in the size towards the basal and apical region of the rachis. Leaflet is rough with midrib and lateral veins. It shows dichotomous branching. Bifurcate the tips near the margin. Venation is dichotomous venation. Younger leaves show circinate venation. Sporangia are formed in group which are called as a sori. A sorus is developed from a pad of tissue known as receptacle, or it is also called as a placenta. On the lower surface near to the margin of leaflet of sporophyll, these spores are present in form of the sori. We will learn about the anatomy of rhizome. Anatomy means study of the internal tissue structures. In TS of rhizome, the shape of TS of rhizome is oval in outline and it shows epidermis, cortex and stele. Epidermis is single layered with quadrangular cells and covered by thick cuticle. Cortex is multi-layered, 4 to 5 layered sclerenchymatous hypodermis and inner multi-layer broad parenchymatous region is present. Parenchymatous region has root traces and leaf traces. Remnants of leaf or root is called as a traces. Root traces and leaf traces are present in the cortex. Then stellar structure differs from species to species. In Pteris vittata, it is dictyostele with a ring of fascicular strands. Meristili are embedded in the parenchymatous ground tissue. Pericycle is present below the epidermis and surrounding to the phloem. Phloem has only sieve cells and phloem parenchyma. It completely surrounds the xylem. Xylem is present at the center of meristili. It shows central protoxylem and it shows tracheids and xylem parenchyma. Then pericycle is present near the endodermis. It is the part of stellar system. See this figure, anatomy of rhizome, which shows cuticle, epidermis, parenchymatous cortex, metaxylem, endodermis region, pericycle region. The next section is anatomy of rachis. It is semicircular in outline. It shows epidermis, ground tissue and vascular bundle. Epidermis is single layered with quadrangular cells covered by thick cuticle. Some cells give rise uniserate bicellular hairs called ramenta. So hair-like structures are present on the epidermis known as a ramenta. Ground tissue are multi-layered. It is with sclerenchymatous hypodermis followed by parenchymatous tissue. Vascular bundle is V-shaped or U-shaped, embedded in the parenchymatous ground tissue. It resembles meristili and has single layered endodermis with Casparian strips. Pericycle is 1 to 2 layered and parenchymatous. Xylem is present at center surrounded by phloem. So simple type of protostele is present in this rachis or petiole. Rachis is also called as a petiole structure. See this figure, TS of petiole or rachis showing different parts. You know about the different parts: ramenta, cuticle, epidermis, hypodermis, cortex region, endodermis, pericycle, phloem, xylem are present in the vascular tissue or in the vascular bundle. The next part is anatomy of the leaflets. It has epidermis, mesophyll and vascular bundle. Epidermis is single layered, present on both upper and lower surface. It has stomata on lower epidermis. Mesophyll is homogeneous or differentiated into upper palisade and lower spongy tissue with broad intercellular space. Hypodermal region of midrib has sclerenchymatous strips in abaxial and adaxial side. That is, on upper and lower surface, sclerenchymatous strips are present in the midrib region. Midrib has single concentric amphicribal vascular bundle. Xylem is surrounded by phloem. Pericycle and endodermis are present in the vascular bundle. These are single layered structures. Vascular strands are embedded in the mesophyll region. This figure represents outline of TS of leaflet, then TS of fertile leaflet showing internal structures, cuticle, mesophyll, air space, vascular strands. Stomata are present on the lower surface only. Sporangium are present. Receptacle is the structure containing sporangia. Then in vascular bundle, endodermis, pericycle, phloem and xylem, these tissues are present. This is vertical section of fertile pinna showing marginal sorus on each side of the lamina. It is covered by reflexed margin. The structure containing sporangia is called as a receptacle. On both sides of the leaflet, these sporangium structures are present, which are fertile structure. File section consists of vascular bundle at the center. In this slide, we will study about the structure of sporangium. Sporangium shows leptosporangiate development because it is developed from a single initial. It has a multicellular long slender stalk with pedicel or seta and globose oval body, which is known as a capsule or spore sac. Spore sac has single layered jacket structure. Some cells of jacket are modified to form the annulus and stomium. Annulus is ring-like structure with vertically elongated cell. It begins from the stalk on one side, encircles the top and extends up to half of the sac. Interior to the jacket, 1 to 2 layered nutritive tissue are present. This layer is called as a tapetum. So tapetum includes nutritive tissues. It encircles the sporogenous tissue. Sporogenous tissue forms 12 spore mother cells that divide meiotically to form 48 haploids. Morphologically, similar spores are present in spore sac. So, condition is homosporous condition. Young sporangium is green and mature sporangium is dark brown in color. With the dryness of annulus, sporangial all breakdown takes place and spores are liberated in air. Spores are unicellular, reddish brown subglobose and have clear triradiate mark known as trility on its surface. Sporole has outer thick sculptured exine and inner thin smooth intine. That is, ornamentation is present on the outer thick exine. It is with design. Sculptured means with design. This figure represents structure and dehiscence of sporangium in Pteris vittata. See the structure of spores. Sporangium is with annulus. It is with stomium. Then spores are liberated. Each spore is with exine, intine and nucleus. The next part is reproduction. Reproduction in the Pteris takes place by vegetative method and sexual reproduction method. By breakdown of rhizome, rhizome pieces are formed and new plant is developed from this rhizome. This is vegetative method of reproduction. The next part is sexual reproduction in Pteris. It started with germination of spore. Antheridia are present in sexual reproduction. These are the male sex organs, which are small sessile and globular structures, consist of 3 tabular cell jackets. These are first ring cell that covers the basal portion of antheridial body, second ring cell that covers the middle portion of antheridial body, and cap cell that covers the apical portion of the antheridium. The cap cell forms the lead-like structure. It is covering-like structure. Cap cell forms covering-like structure. Apart from these cells, there is a fourth cell at the base that can be considered as a single cell stalk. The all encloses 32 spermatocytes. Each one is metamorphosed into long spiral coiled multiciliate motile anthrozoite. So spermatocytes forms anthrozoites. Dehiscence of mature antheridium takes place in the presence of water. Jacket cells and androcytes absorb water, swell and get open or burst. Structure of Archegonia. These are developed later than antheridia near to apical notch of the prothalas. It is flask shaped with swollen embedded venter in prothalas and projected short, slender, slightly curved neck. Neck consists of single layered jacket with 4 vertical rows of cells, with each row having 4 cells. So the total number of jacket cells is 16. Opercular cells or lead cells are absent in the Archegonia. Interior to jacket of neck, a single dikaryotic neck canal cell is present. Then about the structure of Prothalas. It is heart-shaped, dorsiventral, short-lived, anterior-posterior dependent gametophyte. It is independent gametophyte structure. It grows in moist shady habitats. It is located near notch. Prothalas is green, small, flat, thin. Thallus is autotrophic in nature. Itself, it produces food material. It is green in color. Possible to do the function of photosynthesis. It is autotrophic structure. It has a deep U-shaped apical notch. It is thick cushion like structure with many celled. These cells are thick at the center and gradually becomes thin towards the margin. It is single celled in thickness at the margin. So this is about Prothalas structure. It is parenchymatous cells without intercellular space. It consists of large vacuole. Peripheral cytoplasm has a haploid nucleus and discoid chloroplast. Brown, delicate hairs, thin-walled unicellular rhizoids are present in the prothalas. It is usually monoecious. The sex organs are developed on ventral surface and exposed directly to moisture. Male reproductive organ Antheridia, female reproductive organ Archegonia. Antheridia develop earlier than the female reproductive structures Archegonia. Hence, Prothalas is called as a protandrous. Protandrous, Prothalas. See the structure of mature Prothalas containing Archegonia and Antheridia. It also shows rhizoids. Reproductive structures are Antheridia and Archegonia. Archegonia are produced in cushion-like structure in the Prothalas. The mature Prothalas of Pteris is heart-shaped structure containing the sex organs Antheridia and Archegonia, which are male reproductive structures and female reproductive structures. Then during the fertilization, mature male gametes swim towards Archegonium, enters infenter through the neck canal and fertilize egg. Hence diploid zygote is formed. Unicellular zygote develop into sporophyte through successive growth and differentiation. See this figure. Archegonium is with egg cell, Antheridium is with sperm cells or anthrozoids. Each anthrozoid is with flagella structure. It is motile structure. So able to move towards the Archegonium, enters inside the Archegonium, fuse with egg cell and forms the diploid zygote structure. Then new embryo development. Zygote divide to form 32-celled embryo. Hypobasal cell forms stem, apex and foot. Epibasal cell produces cotyledon and root. The venter of Archegonium forms a protective layer calyptra around the embryo. The root forms Prothalas, which develop into sporling. These sporelings forms first leaf. So like this there is a development of embryo into the plant. This figure represents stages of Archegonium development and embryo development. So zygote forms epibasal cell, hypobasal cell. Then it develops stem, foot and leaf. There is growth of stem, foot and leaf structure in the structure of Prothalas. Then it forms the first leaf. First leaf is formed and plant of Pteris will start to develop from the plantlet stage. First plantlets are formed, then entire Pteris plant is formed. This slide represents life cycle. In the life cycle of Pteris, spores germinate, forms gametophyte structure, male reproductive structure Antheridium, female reproductive structure Archegonium is formed. Then spermatozoids and egg are formed. These are formed in Antheridium and Archegonium respectively. After fertilization produce zygote. Zygote will form sporophytic structure with embryo. Then sporangium and spore mother cell are formed in the sporophytic structure. Spore mother cell again produce the spores. Like this cycle is repeated. Cycle shows gametophytic and sporophytic structures in which spores are produced, zygote is produced. Sporophytic structure shows presence of sporangium and spores, spore mother cells. Like this Pteris life cycle is represented in this diagram. Thank you.